Hi guys, so let's start another chapter, another unit, chapter 8. And chapter 8, this whole unit's about genetics, right? And how basically hereditary molecules like DNA, how it gets passed on to other cells, how it gets passed to our offspring, etc. And before we start in any of that, we have to talk about cellular reproduction, so how cells come from other cells, right? And this is actually part of the cell theory, cells coming from other cells. So we're going to start with chapter 8. I believe we go into chapter 9 and 10, and that is the end of this unit. So we are all born with a specific genome, right? Our genome is basically the complete collection of our DNA, so are the complete collection of our genetic information. So basically our genome is all of our DNA. Each one of our cells has our entire genome. Okay, so every single one of our cells, except some few exceptions, have our entire DNA code in them. In the genome, in our genome, it exists as units called genes. Okay, so these genes lie on the DNA. So your DNA is composed of a lot of genes. In fact, humans have about 25,000 genes, but then the rest of the DNA is actually not genes, right? And we like to call it junk DNA, even though it's really not junk, but we just didn't know what to call it, so we just call it junk DNA. But we have these genes on our DNA, and it is those genes that code specific kinds of proteins. And we'll look into future chapters in this section that show you how these proteins come about from the genes, right? So living things develop and function each day through the information provided by genes in the genome. Like I said, your genome and your genes, those are your code, that it codes what you are. And the information for all the production of all the proteins is contained in DNA, because genes are going to code for all the proteins that make you who you are. And so at the end of the story, DNA codes for you, because it codes for all the proteins that make you you, right? So all the cells have the entirety of their genome in their nucleus as DNA. And then some cells, cells tend to express specific parts of that DNA of that genome. What I mean by expressing, that means express, which says right here, I'm going to highlight it, I'm going to make it in bold right here. Express just means that the gene is then made into the protein that it then encodes, right? So the thing is, though, every single cell in our body, except some few exceptions, right, has the entire genome in that cell. But the thing is, your liver cells are not going to want to make any kind of proteins that are important for your brain, because your liver cells want to make proteins that are important for your liver. Does that make sense? And so because of this, each cell chooses to express and therefore make into proteins specific kind of genes. So your liver cells are only going to express liver genes and make them, by expressing them, they'll make them into liver proteins. Your brain cells, your nerve cells, they're going to only express from the entire genome that they have, right? They're only going to express whatever nerve brain protein that is encoded by the brain nerve genes, if that makes sense. So different genes are active in different cells, and this is why we have different cells, because different genes are active in them. So this is what distinguishes one cell from another. Okay, so this is just a little bit of a rundown about the genome. And so now that we know what genomes are, we can start looking into what cells are, right? And how they replicate, how they duplicate. So cells need to divide or duplicate. It's actually really interesting. Biology is the only place, I think, in all of the English language and in the entire world where division and multiplication means the same thing. Because when a cell divides, it divides into two, and now we have two. So it's technically multiplied. So division and multiplication in biology mean the same thing. So cells need to divide or multiply or duplicate. This is important because this is how we carry on life. This attributes to the continuity of life, right? If cells stop dividing, that means you don't have cells that are specifically made to be useful for sex, right? So you can't have kids anymore. You can't have offspring. This is not just with humans. This is with any other kind of animal. Other kinds of organisms, as we'll see, they need to divide their cells in order to make more offspring. It's just how it is. So we need cells to divide in order to actually allow for the continuity of life. So cell division, which should also be in bold, is simply the reproduction of a cell. So how a cell makes a copy of itself. Okay, and when a cell makes a copy of itself, so this is a cell, and it's going to divide through cellular division to reproduce and we have two cells. These two cells that come about from that original parent cell, those two cells are called daughter cells. So the daughter cells result from cell division, and these daughter cells are going to be identical to each other, and they're going to be, obviously, since they're identical to each other, they're also identical to the parent cell. And we'll see how that happens a little bit later on. So prior to any kind of cellular division, the first thing that has to happen is that your DNA needs to be duplicated. So your chromosomes, and you guys know that your DNA is found on chromosomes, your chromosomes need to duplicate. And then this is the reason why you get two cells in the first place. Because if your chromosomes duplicate, right, then the two chromosomes or the two DNA copies that you make can go to opposite poles of the cell, and then the cell can pinch in, and now we have two cells, each with the same kind of DNA, and that's the way they're genetically identical. So I'm going to try and show this by using a Sharpie cap. Okay, this Sharpie cap is going to be my DNA, and it's found within my cell. My cell is my hands right here, right? So now the cell is going about through daily life, blah, blah, blah. I lost my DNA. Okay, the cell is going about through daily life. And now at a certain point in the cell's life, it's going to need to have to divide. It's going to have to divide. But remember, prior to division, what has to happen before a parent cell splits into two, prior to division, it has to duplicate its chromosomes. So it's going to duplicate, it's going to replicate this and make another one that is identical to it. Okay, absolutely identical. They hold the same, it's basically, so now instead of having one genome, we have two genomes in one cell. Okay, they have the same genes. This DNA copy is identical to this DNA copy. At this point, then we have cellular division. So you can see how my hands are pinching in. Cellular division, and we have two cells. And since both of these cells have identical DNAs, right, to each other, they are genetically identical. They're clones of themselves, of each other. And so they carry out the same exact thing. And this is very, very important, right? So cellular division has to happen after our chromosomes or DNA gets replicated. So cellular division is very important because it actually serves a lot of different roles. First off, it's very, very important because it maintains and serves the body, right? So you have cells that are dying, right? You have cells that die. Your skin cells die all the time. In fact, I don't know if you guys know this, but a lot of the dust in your house is actually mainly from dead skin cells. So your skin cells die all the time. Other parts of your cells, other cells in your body also die. But when they die, they have to be replaced. That would be a very tragic world if all our cells did was die. They need to be replaced. They can also grow, right? Cells themselves also grow to an extent, but they can't go too big. Once they arrive at a certain point, they're going to have to divide just to maintain that same size that all cells have. And a lot of times also in certain stages of your life, you have to create more cells. So you don't look anything like you looked like when you were a baby, right? When you were a baby, now going from baby to adult, the reason why you look so different is because you've made more cells. You have grown, especially things in like teenagers. You guys have probably noticed in teenagers, a lot of male teenagers, when like it seems like over the course of a summer, they come back from summer break and all of a sudden they're really super tall, right? It happens. That's because their bone cells have divided. Their bone cells have replicated more, and therefore they are now taller. It's also needed for reproduction, okay? So certain types of organisms, not humans, but other organisms, specifically single-celled organisms especially, they do not have life partners. They don't have life partners that they meet and then go through sexual intercourse. What they do is they simply divide into, boom, and now we have two instead of one. That's all they do. So that's how they reproduce, simply by dividing into two. This is known as asexual reproduction. And when they do this, they basically make clones of themselves, because the offspring, the daughter cells that come about from them, are clones of each other. So they just make clones of themselves. So with asexual reproduction, there is no sperm, there is no egg. That does not happen. With sexual reproduction, there is the sperm and the egg basically have to meet. Okay. We also have certain kinds. So I said predominantly it is like single-celled organisms that go through asexual reproduction, but also some multicellular organisms can go through asexual reproduction as well, like sea stars, for example. If you chop off the the arm of a sea star, it will grow back, right? Or a lizard, if you chop off the tail of the lizard, it will grow back. That is basically, it grows back because they're making cells to grow themselves, to substitute or take the place of that missing limb or whatever, right? So it's really important. Another thing that I think I mentioned, but I'm just going to expand on it a little bit more, but cells need to be replaced. Even like if they don't die, how about the fact if you hurt yourself, like see here, I actually hurt myself. Oh, you can't see, it's in a very weird spot. Okay, whatever. So yeah, I fall because I'm a klutz, right? And I fall and I have a severe wound right here. If my cells didn't replicate, the wound would never get patched up. So it's important that my cells replicate. Okay, so it's really important for cellular division. Okay, this type of cellular division that we've been talking about, where the cells they divide and they make clones of themselves, this particular cell division is known as mitosis. Okay, so this is known as mitosis. So in fact, here I'm going to write mitosis is important because, okay, so mitosis. Another kind of cellular division we have, and there's only two that you need to know, okay, mitosis, and then the other kind that you need to know is called meiosis. So meiosis is also very important because with meiosis, this particular cellular division actually results in sex cells. So meiosis is the reason why males have sperm cells and females have egg cells. If we couldn't go through meiosis, neither males nor females could actually make these sex cells. Another way to call these reproductive or sex cells is also known as gametes. Okay, so I may be referring to them as gametes from here on out, but I'll try to say egg and sperm just because I feel like that brings the point across better. Okay, so meiosis makes gametes, the egg and the sperm, and this is really important because without the egg and the sperm, we would not have sexual reproduction. Okay, and so without sexual reproduction, we wouldn't have the continuity of life, essentially, because we wouldn't have any way, at least us, we wouldn't have any way of making more offspring, right? Some organisms, like single-celled organisms, like amoebas and bacteria, they basically go through asexual reproduction, okay, where they don't need an egg and a sperm, which we saw here is mitosis actually plays a really big part in that. But for most multicellular organisms, especially animals, we require sexual reproduction to actually make our offspring, and that only comes about with meiosis, because when meiosis divides, it actually makes these gametes. And the gametes that come about from meiosis are not clones of themselves. So this is one of the biggest differences between meiosis and mitosis, is that the gametes are not clones of themselves. They are actually different from one another. So all like the sperm cells that a male could have, each and every single sperm cell is different, okay? So they're all different from each other. In addition, gametes only have half of the parent's DNA. I'm underlining this because this is really important. They only have half of the parent's DNA. And this is very, very important, right? Because hold on, I'm going to change my video and show this. Okay, if meiosis never halved its DNA, okay, so say I have, I now have, this is my adult, and I have 4 pieces of DNA here, okay, and this is fully adult, all right? This is a normal cell and an adult. But with meiosis, we saw that the gamete cells that come about, the sex cells that come about, have half of that DNA. And so that would mean that I would get 2 here and 2 here. Actually, I actually get 4, but we won't get into that just yet. Okay, so these are going to be my gametes. Specifically, I'm going to make these sperm cells. So there's my little tail, my little flagellum, to make them sperm cells. Okay, this is important because once the sperm cell meets an egg, okay, and that's known as fertilization and basically sexual intercourse, the egg also has 2 because it came through meiosis, right? It came about and it halved from 4. We now have 2 chromosomes. Okay, so the DNA was halved in 2. So when the egg and the sperm, when they fuse together, guess what's going to come about? It's going to come about, we're going to have a fetus, a zygote, actually, with 1, 2, 3, 4 from the DNA from the dad and the DNA from the mom. And in total, they have 4 chromosomes. So this is how it works with normal life. If in meiosis we did not have the DNA, this would be very, very problematic. So I'm going to move this over here. If we didn't have the DNA, it would be very bad. So if we had a dude that had 4 chromosomes in it, and meiosis happened without having the DNA, which remember does not occur, we'd have 4 here and then 4 here. And let's make this sperm, okay? And then we'd had another female, like a female would also had 4, and she went through meiosis, and she also did not go through the correct meiosis, and she didn't have the DNA. So this means that when the sperm and the egg come together, we now have a zygote with 8, 1, 2, 3, 4 from the mom, and 1, 2, 3, 4 from the dad. 8 chromosomes. This would be bad, right? And if we continue this way, if meiosis happens again, and again the DNA is not halved, the sperm would have 8 cells, and then it would come together with an egg that had 8 cells, and 8 chromosomes, I meant to say, sorry, the sperm had 8 chromosomes, and then it will come together with an egg that had 8 chromosomes, and together they make a baby with 16 chromosomes. So if this did not happen, our DNA would basically would increase exponentially, right? And this would, and it's not good. This would not, this doesn't bring about proper life. And so thankfully this doesn't happen because, as you can see here on the slide, the meiosis allows for the halving of the parent's DNA. And so you only get half of the amount of the parent's DNA in the gametes. Our sex cells have half, our eggs and our sperm cells, our gametes have half of the amount of DNA. So we have 2 kinds of cellular division. We have mitosis and we have meiosis. Mitosis is important for the growth and maintenance of multicellular organisms and also for asexual reproduction, and meiosis is important for making gametes. Please take note of the purposes of these kinds of cellular division because they are very different from each other. And speaking about that, you have a bio write-up that is due on the day or the day before your exam, okay? Your bio write-up around the day of your exam. So look over coursework to see when it's due. This bio write-up is going to be on mitosis and meiosis, where I want you to explain mitosis, I want you to explain meiosis, and then compare them, compare and contrast them, and see where they're different. The main one of the differences, not the only, one of the differences is in the roles that they carry out, right? And this is something that I'm going to be looking to see if you actually include the roles of mitosis and the roles of meiosis are very, very different. Mitosis is there for replenishing dead cells, replenishing wounds, right? For growth of the cells, for growth of the living organism. Like this is how we go from teenager to really, really tall teenager and then adult. And then it's also important for asexual reproduction because it simply makes clones of itself. Meiosis is important for sexual reproduction because it makes the cells that are involved in sexual reproduction in the first place, it makes the gametes. Okay. So now that we know what the 2 types of cellular division are and that before every single type of cellular division, our DNA has to be replicated, we can move on and look into the cell cycle and look a little bit more into mitosis. I was like, which one is it? Mitosis. So just a recap, just to let you guys know a little bit more about DNA. In a eukaryotic cell, all of our DNA is found on chromosomes, and all of these chromosomes are found within the nucleus of the eukaryotic cell. Remember, eukaryotic cells have a true nucleus. Each eukaryotic chromosome contains very long DNA molecules, and these DNA molecules have genes on, literally lying on them, okay? Parts of the DNA encode for specific kinds of genes. And remember, humans have about 25,000 genes. The number of chromosomes in a eukaryotic cell depends on this number, on the actual species. So humans, you've probably heard of this, we have 23 pairs of chromosomes. 23 pairs, so 23 times 2, that's 46 chromosomes. There's other things out there like koalas have 16 chromosomes. A rat, that's disgusting. This particular rat has 102 chromosomes. So a lot of people think that the more chromosomes you have, the more complex you are. That's not necessarily the case. I think actually there's even a species of butterfly that has about 256 chromosomes. So it obviously is not the case. So again, a chromosome is just a unit of where you find your DNA. Your DNA is found and kind of condensed in this chromosome. It's a structural unit containing part or all of an organism's genome, consisting of DNA and its associated proteins. So, and then the collection of all these chromosomes are going to make up your genome, are going to be the whole entire DNA that makes you who you are. So all of those 23 pairs of chromosomes, all of those chromosomes make up our genome, okay? So in order for our DNA, remember how I said the nucleus holds our chromosomes, the nucleus holds our DNA. In order for the DNA to actually be found within our nucleus, a lot of things have to happen. Because if I take the DNA from just one of my cells, I can extend it and it'll be about 6 feet long. So actually even, yeah, about 6 feet long. So really, really long. And yet that entire long thing has to somehow fit within a very tiny structure in your very, very tiny cells. So how does this happen? So this happens through basically the use of proteins. So if this is my DNA, see how it's, I'm using my headphones here, see how it's really, really long, right? And yet it has to fit inside my inside my nucleus. So the way it occurs is that you have proteins, which are going to be my finger, proteins, and you can see these proteins right here. They're known as histones, okay? Histones, okay, little stones, okay? These little histones are what allow the DNA to wrap around. It allows the DNA to wrap around the histones, and you can see that that really long DNA, which was my headphones, now gets really nice and condensed in one little thing. So it's like a string and spool effect. It basically spools the DNA around it. We spool the DNA around it, and it packages it and condenses my DNA into a nice little spot, a nice little thing, and that's how it can go in and fit inside my nucleus, right? And so we have all this crazy packaging and condensing until the DNA starts winding around the histones, right? This interaction, the interaction between the DNA and the protein is known as chromatin. You can find it on this slide right here. Is just the interaction, and so therefore what chromosomes are made of, because if it's important, right? If DNA didn't have anything to wound itself around, it would just be like a jarbled mess. So, and just too big to fit into our nucleus. So it has to wind itself around histones or proteins, and this is known as chromatin, and this forms our chromosome, okay? The histones then package themselves up a little bit more, and they form nucleosomes. So nucleosomes are just little beads of like DNA and protein, and then these nucleosomes keep like kind of they kind of coil up on each other, and you can see that it becomes this really tight-knit structure of a lot of DNA, and this is known as a chromosome. So this part right here is known as a chromosome. Why are you telling us this, Alessia? This is really important because your DNA has to be made into these really tightly supercoiled condensed chromosome formats in order for you to actually go ahead and divide your cell, okay? And I'll explain that in just a second. So if you see here, like remember, so before you divide your cell, chromosomes have to condense and tighten up, and basically your DNA has to coil up like so. So when you look at it under a microscope, you can actually see these things come about. Like you could, these little worms, of like these purple worms are not worms, they're actually DNA. And it's so condensed and so like packaged up that you can see them under the microscope, when normally you wouldn't be able to see DNA if it was just like kind of unraveled, okay? So right before any kind of cellular division, you can see your DNA is packaged up like this. So this is what it looks uncondensed. This is the uncondensed form of DNA. And this is the condensed form of DNA. This is the chromosomes. You see how different it is? It's in all the DNA that you find in this chromosome is way more than way more than the DNA found here. This looks like a spaghetti mess. It looks like what was the thing called? The party string? I forgot what it's called. Anyways, never mind. It looks like a spaghetti mess, right? And here you have these nice knit structures. This is very important. I want you to keep this in the back of your mind as we progress through mitosis, as we go into mitosis soon, okay? So your DNA is usually found in this condensed form, but before mitosis happens, it's made into this really condensed chromosome format, and that's when we can see it under the microscope, okay? There's a nice little video for you guys to watch, so please go watch that video before we at a point in your time, in your week, in your day. Okay, so Now that we know what DNA looks like and how it has to be condensed before mitosis, we also have to talk about the fact that we have to actually duplicate our DNA. Okay, duplicate our DNA, because remember, prior to cellular division, we duplicate our DNA, red cap, second red cap, and that DNA then gets separated and brought to opposite sides of the poles of the cell, opposite sides of the cell, and then the cell pinches in and we have two cells. All right, so boom, boom, boom. Okay, so we have to duplicate our DNA. So again, this is in bold and capitalized. DNA replicates prior to cellular division. This could come up in your exam. Hint, hint, wink, wink. All right. So this is important because in this way, each cell has all the genome in it. If this wasn't the case, the cell would not have all the genome in it. If this was not divided, the two cells that would come about from it would have half of that DNA and half of the other DNA, and that's not how it works with mitosis. With mitosis, we want the full genome effect, right? So before a cell begins a division process, the DNA molecule of each chromosome is copied through the process of DNA replication. And then we have the whole new histones are added to kind of repackage everything up again. So at the end of when our chromosome gets duplicated, we actually form a duplicated chromosome. And since these two strands, these two different strands of DNA are identical to each other, we call these sister chromatids. Okay? So in my weird hand example, this would be my DNA. Oh my gosh, I can't do this. This would be my DNA. All right. When we duplicate it, this one is identical to this first red cap, to this first DNA molecule, and so they are going to be technically joined together. And in addition, they're clones of each other, so they're called sister chromatids. And so now the next part is to kind of separate these sister chromatids apart from each other to make them go to opposite poles of the cell. Imagine there's a cell around this. Okay, I can't make my hands do that. So imagine there's a cell around the two DNAs, the two sister chromatids that make up the duplicated chromosome, one duplicated chromosome. And then they are going to be separated from each other, go to opposite poles of the cell, and then in the middle, the cell is going to pinch in and make two daughter cells. Okay. Another thing that you may have noticed is that these sister chromatids have a waist, okay, or chromosomes have a waist. This little waist, or this little cinching in narrow waist, okay, this part where it cinches in, this is known as a centromere. And this is important because this is what, in order to pry these sister chromatids away from each other, something needs to latch on. And the place where it latches on is on that centromere, okay? So now that we know that DNA has to be replicated, it makes sense. This is what I was trying to show you guys with my little weird finger Sharpie cap scenario, okay, and hand scenario, but this is what's actually happening. This is a cell with a DNA, right, with one copy of DNA. This cell has to go through cellular division. What must happen prior to cellular division, any kind of cellular division, either mitosis or meiosis, the DNA has to replicate. So one DNA molecule, and after DNA replication, two DNA molecules. Come mitosis or even meiosis, but in this case, we're focusing on mitosis right now because it's the simplest one, right? Come mitosis, the DNA gets partitioned, it gets segregated, and it gets pulled to opposite poles of the cell. So this would be one sister chromatid going to one side of the cell and the other sister chromatid going to the other side of the cell. And then at the end, you see how this elongated cell is kind of already pinching in the middle. This pinching in continues until plop, we have two cells, each with the DNA, right? This DNA is identical to the parent DNA. This DNA is also identical to the parent DNA. And the DNA between themselves are identical to each other. So they are clones of the parent and clones of themselves. And so with mitosis, we have two identical quantities of DNA that get apportioned off to two daughter cells. And so the two daughter cells are identical to each other. Okay, so we go from an unduplicated chromosome. After DNA replication, we have a duplicated chromosome. And so you see each in this duplicated chromosome, this is a sister chromatid and this is another sister chromatid. And so we have to pry them apart from each other. Okay? So now that we know the basis of everything, we can actually look into the cell cycle. So the cell cycle is basically the cycle and the lifespan of a cell. It's basically the circle of life. If you feel free to sing the Lion King song right here, so please. This is what every time I think of the cell cycle, I start thinking the circle of life, right? I start thinking of that. This is basically what it is. because every single one of your cell is born at a certain point, and at a certain point, it's also going to die, or it's going to duplicate, right? And so this is what the cell cycle is. It is the ordered sequence of events that extends from the time a cell is first formed, so the cell is first born from dividing parent, to its own division into two cells, okay? And there's specific stages within the cell cycle. And I'm going to draw them out for you right now. I'm going to get another piece of paper. Okay, so there's specific phases in the cell cycle. I'm going to draw it out for you so you can't see the PowerPoint, but all of this information is found in the PowerPoints right here, actually. Okay. Boom. Here we go. So I'm going to take another one. Okay, so our cell is born from a parent cell, right? So this is our cell. This is going to eventually focus. Hold on. Take our time. Don't worry, we have all the time. No, we don't actually have a lot of time to lose. Come on. Hopefully it'll focus. But you can see this is my cell, okay? I'm not going to draw anything in the middle. This is a cell. And in this cell, we have I'm going to draw one molecule of DNA in there. I'm just going to call it DNA, okay? This cell, as soon as it's born, it's just going to carry out its normal everyday life. It's going to just do whatever it has to do. It's going to carry out its normal daily, there we go, its normal daily activities. When it carries out its normal daily activities, this particular cell is found in the G1, we call it the G1 phase, okay? G1 phase. The G stands for gap, okay? But it's not really a gap. There's still things happening in this gap, in this phase, where the cell carries out whatever it's supposed to do. So if it's a liver cell, it's going to participate and contribute to the liver organ, to whatever the liver has to do, right? So this is the cell carrying out its normal daily activities, okay? At a certain point, let's say I hurt myself, right? I hurt myself and I need to make more cells. So I get a signal, Alessia gets hurt, I need to make more cells, so I need to go through cellular division. You guys already know this. What must happen prior to cellular division? I'm gonna give you a couple of seconds. So what happens prior to cellular division? Prior to cellular division, we have to replicate our DNA. So right now we are about to start dividing our cell, but before we do that, right, because Alessia got hurt right here, we need to replenish our cells because I'm like, I have a wound or something like that. So prior to that, we have to replicate our DNA. So we enter the cell in the cell cycle. So this is the entire cell cycle, okay? The entire cell cycle. I'm not done yet. I haven't started from the beginning. I haven't gone to the beginning yet, right? There's a phase in the cell cycle called the S phase. The S stands for synthesis, because this is where we synthesize and make our DNA. So we replicate our DNA. So at the end of the S phase, our cell looks something like this, with 2 molecules of DNA, okay? So now that we've replicated our DNA, we can start going through the actual cellular division, in this case, mitosis. But we still need a little bit of time. The process of mitosis, as you can imagine, is very invasive. Like the cell is literally dividing into 2. It's squeezing, it's in, like the middle of the cell gets squeezed in and it plops into 2. It's a very invasive process. So there's actually a stage prior to cellular division called the G2 phase, where the cell preps itself for this particular invasive procedure, right? As you would prep yourself for an invasive procedure, the cell does the same thing. Phase, I wrote phage, sorry. The G2 phase, okay? This G2 phase, the cell prepares for division. So if it were me, if I was a cell and I'd have to prepare for this really crazy, you know, kind of like, it looks like there's a lot of exercise. It's very like active, right? Very active thing. I would hype myself up by listening to some really cool music, right? So I would listen to something like Eye of a Tiger. And that is exactly what the cell is doing. It is hyping itself up. It is preparing all the specific proteins that it needs in order to actually go through cellular division. So we need this G2 phase. It's a very important phase, G2 phase. At the end of G2 phase, Then we have division, cellular division, which is called meiosis. No, sorry. Oh my goodness. It is one type of is called meiosis, but in this case right here, we're focusing first on mitosis. Okay, so during this period right here, I'm going to make it red. Okay, during this period right here, we go through mitosis. And at the end of mitosis, the cell divides into two. We get one cell and we get another cell. And then guess what happens to those two cells? This cell goes into cellular division, the rest of the cell cycle. Sorry, this cell continues with the rest of the cell cycle. That's what I meant to say. And this cell as well continues with its own cell cycle. This cell will also go into G1. Eventually, it will go into the S phase, and then the G2 phase, and then again, the cellular division, mitosis. So these are the phases of the cell cycle. Another thing that I want to mention is that the G1, the S phase, and the G2 all make up what we call the interphase stage. Okay, so interphase is G1 plus S phase plus the G2 phase. Okay, so if I find myself, if myself is in myself, I meant to keep saying my cell. If my cell is in the S phase, that cell is going through DNA replication, and it's also an interphase. The only time my cell is not in interphase is when it starts mitosis. Okay, so cellular division is not part of interphase. G1 is interphase, S phase is interphase, G2 is interphase. Mitosis is not interphase. And this is the normal cell cycle. So as you can see, the cell is born, the cell carries out its normal daily activities. Once the cell knows that it has to divide, prior to division, it's going to synthesize and replicate its DNA. Then it's going to basically prepare itself for that invasive process of cellular division, and then it's going to actually go through division, go through mitosis, and split into two. And once it does that, each daughter cell is going to start into the cell cycle all over again. Okay, and this is what you see here. This is what you see basically in this slide right here, and in the next slide, and so forth. You talk about interphase and what that is. Okay, you talk about the mitotic phase or the mitosis and what that is. This includes mitosis and cytokinesis. We'll talk about that in just a second. And that's how we go through cellular division. Okay, and again here you have, it talks about interphase, how interphase is divided into three subphases: G1, S phase, and the G2 phase. All right, so now that we know what the cell cycle is, we can actually look into what mitosis is. So this is when we're honing into the actual process of cellular division and what happens there. Okay, so the mitotic phase includes two overlapping processes. First off, we have mitosis, right, which is where we actually separate our DNA. So if you look back at the screen at what I'm drawing here, the two DNA, this would basically in one cell. If I have a cell right here, I know there's things at the back. Sorry, I'm just gonna. This is my cell. I had my DNA. I replicated my DNA. I know it's, I can't cut this off, sorry. But I replicated my DNA, so now I have two copies of DNA, one duplicate chromosome and two sister chromatids. And then during mitosis is simply the actual separation of these two DNA molecules to opposite poles of the cell. And remember, with bigger organisms like us, we have more than one chromosome, right? We have actually 46 of them. This is just an example with two chromosomes. So two sister chromatids coming and separating. Okay, so one chromosome, two sister chromatids. At the end, this is mitosis, is the division of the nuclear component, the division of the chromosomes. And then we have cytokinesis, as you can see here. Cytokinesis, cyto means cell, kinesis means movement. So this is the actual movement of the cell, and the cell is going to be pinching in and separating and pinching in into like so. Wait, hold on. I don't know if I could, like so, and pinching into two. Okay, so this is when the cell actually divides into two. So the combination of mitosis and cytokinesis produces two genetically identical daughter cells, each fully equipped with a nucleus, cytoplasm, organelles, and a plasma membrane. So each of them are basically their own cell, right? And they continue they can start into interphase and continue and do whatever a cell, that particular cell has to do. So mitosis also has a few stages and phases to it. It actually has 4 phases. These phases are known as PMAT: prophase, metaphase, anaphase, and telophase. So if you think about it, there's actually, if you have difficulties remembering PMAT, so prophase, metaphase, anaphase, telophase, one way you can think of it is please make a taco. Okay, please, prophase, make, metaphase, anaphase, taco, telophase. And at the end of all of that, on your taco, you're gonna sprinkle some cilantro. Pew! Okay, kind of like Salt Bae. You're gonna sprinkle cilantro like Salt Bae, and you have cytokinesis. Sorry, you have cytokinesis pretty much at the end, even though cytokinesis can kind of overlap with telophase a little bit. Okay, so let's look at these please make a taco, prophase, metaphase, anaphase, telophase in detail. So prophase is the first step in mitosis. So actually, I want to go back to my drawing right here real quick. Right? So right now, we've gone through the cell cycle. We're honing into the mitosis. And in mitosis, we're going to have prophase, metaphase, anaphase, telophase, and cytokinesis. And at the end of this, we have 2 daughter cells. Okay, so now we can go. So what happens in prophase? There's a few things that happen in prophase, and you're going to need to know all the things that happen in prophase. So again, this is the beginning of the cell. This is the beginning of the cellular division. Okay, this is the beginning of mitosis. So the first thing that happens is that your nuclear membrane, so the nucleus, the envelope around your nucleus, starts disintegrating. This is important because you're literally going to have to move the DNA to opposite poles of the cell. If there's something that's enclosing the DNA in it, it's not going to work. Remember, this is already duplicated DNA because we've already gone through the S phase, right? So at this point, your duplicated DNA is found within the nucleus. When prophase happens, the nucleus starts disintegrating so that the DNA is just found in the cytoplasm. Another thing that happens is that these things known as centrioles right here, which are what form mitotic spindle, they start moving to opposite poles of the cell. Why is this important? What is mitotic spindle? Mitotic spindle are the little fingers that actually attach to, remember my chromosomes, my sister chromatids, something has to pull them away from each other. That's something, those little fingers, those little yellow, like in the picture, they're yellow, okay? Those are the mitotic, that's the mitotic spindle. This is just the stuff that, these filaments basically that attach the chromatids and separate them from each other. And they attach them, remember, at the centromere, at that little waist. So the nuclear envelope begins to break down. The centrioles, which are what make basically the mitotic spindle, they're also known as centrosomes. The centrosomes go to opposite poles of the cell, right? And in addition, the third thing that happens, so 3 things happen in prophase. The third thing is that the chromosomes start to package up and condense and form really condensed packaged things. Because at this point, right before mitosis is happening, our DNA looks like a spaghetti mess. Remember how I told you to keep that in the back of your head? Our DNA looks like this, and it's replicated. So there's a lot of it. Imagine having to precisely and accurately divide and split one DNA molecule from another when it looks like a spaghetti mess. Have you ever tried doing that to a bowl of spaghetti, just kind of dividing it, right? You probably, like partitioning it to one side of the plate and the other, in that you probably broke off a little bit of the spaghetti, and that little broken off fragment of spaghetti went on the other side of the plate, right? If that happens with mitosis, that is really bad. That can cause all sorts of terrible, terrible problems. This happens in any kind of cellular division. This can be really bad for the cell. It can cause cancer. If it happens in meiosis, it can cause all sorts of horrible genetic defects. Okay, so our DNA should not and cannot look like a spaghetti uncoiled, uncondensed mess. It has to be nicely packed chromosomes, because if they're nicely packed, just like these things, if they're nicely packed, you can actually split them and not cause any fragmentation of the DNA. Nothing comes apart, right? So in prophase, our DNA gets condensed into actual nice, cute, pudgy chromosomes. And these cute, pudgy chromosomes can then be separated. So after prophase, PM, please make, comes metaphase. At this point, our chromosomes are nice and pudgy. Okay, our DNA is nice and pudgy. And the mitotic spindle, remember those fingers, they attach to the nice and pudgy chromosomes, duplicated chromosomes. And they attach themselves to the duplicated chromosomes, and they wiggle the chromosomes until all of them are basically found in the middle. This middle, okay, is known as a metaphase plate. It's not an actual feature of the cell. It's just the kind of like the equator of the cell. And just like the equator isn't something tangible, right? Just like the equator, the metaphase plate isn't something tangible either. It's just the middle of the cell, and this is where the chromosomes align. And if you look at each of these chromosomes, one sister chromatid is facing the top, and the other sister chromatid is facing the bottom. So it's going to be really easy to pull these things apart, right? None of them are kind of like diagonal or askew. One's facing the top or one's facing the bottom. Or in other pictures, it could be one's facing the left and one's facing the right. This is good. It shouldn't be diagonal, and it shouldn't be in any other kind of random position in the cell. If they're all along the metaphase plate, all along the middle, it's going to be easier to split them up, right? So in metaphase, meta means middle, guys, okay? Metaphase plate, in metaphase, the chromosomes align to form the metaphase plate in the middle of the cell, and the mitotic spindle are attached to each sister chromatid, specifically at the centromere that's cinched in waist of the sister chromatid. In anaphase, the sister chromatids finally get pulled apart. So the mitotic spindle literally kind of works like a fishing rod. So you know when you go fishing, you wheel in the fish, right? This is exactly what the mitotic spindle is doing. It's wheeling in the sister chromatid that it's attached to. And so this separates the sister chromatids from each other. See here, they were together. Now they are disattached, right? Or unattached. They separate the sister chromatids and they start wheeling it to opposite sides, moving them to opposite poles of the cell. So after anaphase, we have telophase or telophase, depending on how you want to pronounce it. At this point, the chromosomes are opposite sides of the cell. You can see that. They're on opposite sides of the cell. The mitosis is basically coming to an end. So because the duplicate chromosomes have been completely and successfully separated and segregated to go to opposite poles of the cell, our chromosomes can now decondense. And so they become more like a spaghetti mess, right? And we can start forming our nuclear envelope around the chromosomes. And so this is basic. So a nucleus starts reforming around the DNA. And so you can see here at the same time in telophase, a little bit of pinching happens. And this pinching is basically cytokinesis. So the end of the cellular division. So cytokinesis usually occurs during telophase, sometimes even in anaphase. This divides the actual cell. It divides the cytoplasm. And one thing that is important for you to remember is that cytokinesis is different in plants and in animal cells. How and why, Alessia? We'll get to that in just a second. Okay, and maybe you should also kind of think about it yourself. Why do you think cytokinesis is different between animal cells and plant cells? What do plant cells have in terms of features, cellular features, that animal cells do not have? Okay, so let's look at cytokinesis in animal cells. Sorry, cytokinesis in animal cells. So in animal cells, it's really easy. We only have a plasma membrane, basically, that's like the periphery of our cell. And so that plasma membrane starts pinching in. And it gets pinched in by something known as a contractile ring. It starts forming a cleavage furrow, which just means it's like an indentation. And then this pinching in continues until plop, it comes out. So cytokinesis in animals, really simple. It's just the pinching in of the cytoplasm until we have two divided cells, okay? And so now we have two finished daughter cells in the animal, and these two daughter cells can proceed with their cell cycle, and they will enter the interphase, right? They'll enter the G1 part of interphase. In plant cells, cytokinesis is a little bit difficult because plant cells not only have a plasma membrane, but outside of the plasma membrane, they have a very rigid structure. Remember that rigid structure? It's called the cell wall. So they literally have a wall around them. And as much as you want, you cannot bend and pinch in a wall. The walls are very, very strong in plant cells. So plant cells carry out the same steps of mitosis, so prophase, metaphase, anaphase, and telophase, but their cytokinesis, though they carry it out, is a little bit different than animal cells because they have to deal with a cell wall, okay? So instead of pinching, what plant cells do is, if you can see, I'm just gonna make this a little bit bigger so you can see it. plant cells literally start making a cell wall in the middle of the two nuclei that were formed in telophase. So these are the two DNAs that are now surrounded by a nuclear envelope in telophase. At this point, the plant cell starts making the cell wall in the middle. And what it does is it forms a cell plate. It literally sends in little vesicles with all the cell wall material and components inside of them. And then these vesicles fuse with each other to form a cell plate. And then eventually the cell plate keeps growing and growing until it fuses with the top of the cell wall right here and the bottom of the cell wall right here, now making two daughter cells. So so cell walls basically inhibit plant cells from pinching in cytokinesis. Instead, what they have to do, if this was my plant, they literally have to, if this is my cell, they have to make I can't do this. They have to make, no, they have to make a cell wall, a cell plate in the middle of the two nuclei. Okay, so watch this video, really good video. This also right here, this kind of goes over all of the processes of mitosis. So actually, so interphase as well. You can see the condensed, the uncondensed spaghetti stuff. Prophase, our chromosomes start condensing, our nuclear envelope disappears. The mitotic spindle centrosomes start going to opposite poles of the cell. In metaphase, the mitotic spindle is attached to our duplicated chromosomes, and at this point, they're going to start wiggling them to the middle of the cell, and they're going to form the metaphase cell, the metaphase plate. So right here, and then we have anaphase, where our sister chromatids get pulled apart to opposite poles of the cell. In telophase, our DNA molecules are at opposite poles of the cell, so our nuclear envelopes start forming around them, and the DNA starts uncondensing. And then we have cytokinesis, which could either be pinching in animal cells, as you can see here, it starts pinching, or it could be forming a cell plate between the two nuclei in plant cells. So definitely look at these videos, guys. They're very important. They're very really good. This is a small little video that shows you how it works. It's just like it happens in real time. So these little worms right here, that's actually chromosomes.
This newt cell has already started prophase, the first stage of mitosis. Chromosomes have condensed and become clearly visible, contained within the nuclear envelope. In the cytoplasm, the mitotic spindle begins to form during prophase. During prometaphase, the nuclear envelope breaks up. The chromosomes become even shorter and thicker. The microtubules of the mitotic spindle attach to the chromosomes and move them toward the equator of the cell. In metaphase, the chromosomes convene at the metaphase plate, an imaginary plane equidistant from the spindle poles. Anaphase begins when the paired centromeres of each chromosome separate, and the sister chromatids, now full-fledged.
Ooh, look at that separation. Isn't that cool?
The poles of the cell are
that's anaphase.
The cell.
So now they're opposite poles of the cell.
Nuclei form, and the chromosomes become less tightly coiled. Cytokinesis begins during telophase, resulting in two identical daughter cells.
Isn't that cool? So definitely look at all these videos. They're very important. Now, another thing that we have to talk about when we talk about mitosis is cancer, okay? Because cancer is essentially your mitosis going wrong or mitosis being uncontrolled, right? There's your cells are continuously dividing, so much so that you form basically a bunch of cells where they shouldn't be, and this lump of cells is known as a tumor. Okay, so the reason why tumors come about, and there's good tumors, there's not good tumors, that's actually really bad, benign tumors which are not as harmful, and then there's malignant tumors which are very, very bad for you. And so benign tumors can eventually turn into malignant tumors, but usually with benign tumors, they're a lot more easily removed, and they don't really cause that much havoc, while malignant tumors are harder to remove and they cause a lot more problems. So the reason why these tumors come about is simply because we have uncontrolled cell growth, uncontrolled cell increasing, like an increase in the cell's population or the cell's amount, right? This is usually, this is when in your cell cycle, right? In the cell cycle, things are not listened to. Like your cell does not listen to the cell cycle properly. In the cell cycle, what you'll notice is that there's always these little checkpoints around the cell cycle. I'm just going to move to a cell cycle right here. Littered throughout the cell cycle, okay, there's a lot of checkpoints where the cell kind of, as it proceeds through the cell cycle, it stops And like these proteins, I like to think of them as police proteins. If they look at the cell, they make sure that everything is okay, and then they allow the cell to keep going, right? So there's police proteins in your body that tell you, that allow your cells to proceed with cellular division, or they stop your cell from going into cellular division. So say I hurt myself here, right? But when I hurt myself, one of the cells that wants to divide and replenish that, that chunk of skin that I got rid of, that I don't have anymore, one of the cells is actually mutated. It has really bad DNA. I don't want that cell to replicate anymore, right? I don't want that cell to replicate. And so these police proteins will tell my cells to stop, don't replicate, you're not going anywhere. And that's good, because I don't want to make more clones of that mutated cell, right? And the police proteins will allow other cells to replicate and replenish the part that I have to replenish, right? So there's these checkpoints literally throughout the entire cell cycle, with all these police proteins telling the cell cycle to go forward or to go backwards, or basically to stop. So we have all of these cell cycle controls, right? The cell cycle control system consists of specialized proteins, those police proteins within the cell. And these proteins are able to integrate information from the environment, from the surroundings, and tell the cell cycle to stop or go ahead. Now, in cancer, these police proteins are not listened to. Cancer cells do not listen to these police proteins, and they just keep going forward, even though they should not actually divide, right? So if I hurt myself, the police proteins are going to be like, yes, divide, we need to replenish this. But let's pretend I don't hurt myself. I don't hurt myself, and I have a cancerous cell in my body. This cancerous cell is going to divide, even though I don't need it. And this is why tumors form these lumps, these big masses of cells, because there's just uncontrolled cellular division. So cancer is a disease of the cell cycle. Cancer cells do not respond normally to the cell cycle control system, to the police, right? To the police proteins. They divide excessively, and they can invade other tissues of the cell as well, as soon as they become malignant. Okay. In addition, they also don't, since they go through excessive replication, they also don't listen to police proteins when the police proteins tell them, don't replicate, you're mutated. They don't listen to them. So in cancer, mutated cells replicate. Mutated cells are not good for you. The more mutated your cell is, the more genetic changes you have, and the more problems that cell become, like the more problematic that cell becomes. And so that cell literally will mutate so much that it doesn't know what kind of cell it is anymore, and it's going to cause all sorts of horrible problems. That is basically what cancer is, okay? So cancer cells can form tumors, abnormally growing masses or lumps of body cells. If the abnormal cells remain at the original site, so they remain at a specific site, that lump is known as a benign tumor, okay? And usually we can surgically remove these, not easily, but we can try to surgically, for the most part, surgically remove them, unless it's like a tumor of like the blood or something like that, like leukemia or something. Then obviously we can't surgically remove that. But that is still uncontrolled cell growth, right? Malignant tumors are the types of tumors that spread through neighboring tissues. So this is when the cancer keeps replicating, and it keeps replicating mutated cells. And these mutated cells are no longer, like, if it's, say it happens in the liver, the cell is now so mutated that no longer identifies itself as a liver cell, and it will go somewhere else, right, to the brain or whatever. And this is how your cancer spread. And this is known as metastasis, okay, or metastasis. I'm not sure how you guys pronounce it. This is a spread of cancer cells to the other sites of your cell. And this is when, when a person is said to have really bad cancer, is when it's malignant, okay? So what causes the cells to no longer listen to those police proteins, to those cell control systems, right? What causes it? There's a few things that actually cause it, right? Things like smoking, radiation. There's things known as carcinogenics. Carcinogenics are particular products or substances that cause cancer. So these are things that can be found within, like, even acne cream. We have something known as benzoic acid in acne cream. So look at it. Some acne creams don't have it, but they have, that's a carcinogenic. That causes your cells to mutate and to become cancerous. Also things like UV light, right? This is why we protect ourselves. We put sunscreen, because UV light, that's radiation, that can be carcinogenic, that can mutate our cells and make our cells cancerous. Certain kinds of things that we eat, like any kind of preserved meats, right? They have things like called nitrates and nitrites in them, and those are going to be those can cause cancers. Burnt meat, can also cause, like, you know, that charred, not tight, tasty grilled meat, that burnt thing. I love it. It's really bad for you. Very, very bad for you. Smoking, very bad for you. It has this compound known as aldehydes in it, and they can cause cancer. So there's a few things that we can do to actually prevent ourselves from getting cancer, and it's like it all depends on your lifestyle. So it's just lifestyle stages, like lifestyle changes, like not smoking, exercising adequately, avoiding overexposure to the sun, eating a high fiber, low fat diet, because even what we consume can cause cancer, performing self exams, and regularly visiting your doctor to identify tumors early, right? So there are ways that we can prevent cancers from coming about. If a person does have cancer, there's a few ways that we can treat them, hopefully. There's three main ways. First one is surgery if the tumor is solid. Radiation therapy as well, where we purposely try to kill, like through radiation, only the cancerous cells, but so by putting these high energy radiation beams on the cancer cells. This happens, we also kill some normal cells as well. And so this obviously is one of the reasons why people that are going through cancer treatments they look so sickly, and they're just like they lose hair, etc., because we're also killing their normal cells as well as we try to get rid of the cancer cells. Chemotherapy is when the drugs are used. Again, drugs that more and more are more specific. Through research, they specify more and more like cancer cells, but sometimes these drugs can also harm normal cells. Actually, a lot of times they can harm normal cells as well. But these drugs basically disrupt and inhibit cellular division. And so it's like all your cells no longer go through cellular division. And you can see the drastic and the terrible side effects that it could have, right, when a person goes through chemotherapy. So these are usually used to treat cancers that have been spread throughout the rest of your body as well. So that's what it's really important to know. Mitosis, really important, because it's also, if it goes wrong, it leads to cancer. So in the next video, I'll be going over meiosis, and then I believe that is it, and the abnormalities that we can get with meiosis as well when that doesn't work. So I'll see you guys in the next video. Bye, guys.